Okay, let's start this show on the road. This is Dr. Nelson Garais. I'm recording this August 18th, Tuesday, and this is MED110, Anatomy and Physiology 1, 5DAX, which meets on Tuesday mornings. Now, I always do that. What did we go, just as a recap from last week? So what is due today? Task 2, discussion 2, lesson 2. And we're going over the basis of life, all the bits and parts of which make us us. And we went over chemical bonds, the different kinds of chemical bonds. Go over that that PowerPoint. It has some nice pics in there. The videos are also good. Went over chemical bonds, a little bit about the chemistry of water, specifically hydrogen bonds, and also solubility, on how things that are soluble, like salt into water, how water breaks that up. And the really important, the introduction of organic molecules. You know the 4 types of molecules. That's your carbohydrates, fats, also known as lipids, and your proteins and your nucleic acids. And there are nice notes here and here in your task. So water, bonding, macromolecules, the 4 types of macromolecules, and the basic chemical structure of organisms with focus on bonding. Okay, so what are we doing this week? We're going to continue on that path. You know that atoms make molecules, molecules then make macromolecules, macromolecules then make organelles, organelles make cells. And what are organelles? And that's what we're going to be talking about this week. There's this guy, he was an optician. It's back in the day, this guy Leeuwenhoek. He was messing around with an optician. They're the people who make glasses. So he was messing around with lenses, and he found that if you look at just like a little puddle of water, there's things in it. You don't even need a microscope to look at water to know that there's living things in it. And a living thing is composed of organs. Well, your cell is also composed of organs, and that's what we call organelles. So, and we already now know if the cell is sick, then your histology or your tissues will get sick, then the organs will get sick. If the organs get sick, system gets sick, person gets sick. So let's go over what are the 3 parts. Welcome, welcome. Kindly mute. And if you got any questions, you can put it in the chat, and I'll check the chat periodically. So what's due for next week is your concept map on cell structure, organelles, and the function of organelles. And one of the notes here, I forgot which, I think it's not this one here, the Carolina cell structure and function. It's got all of that in there. But let's look at, I'm a visual guy, so let's look at pictures. That's the best way to look at things. And let's pick, where's the one that I always like to look at? It's 3D. Here you go. Let's look at this. And let's look at the cell from the, I think the best way to do it is this. Let me put it on a word so we can all see it. Okay, so this is your typical cell, or also known as your eukaryotic cell. And if you recall from your medical terminology, U means EU, the prefix means normal or the rule. So this is pretty much the rule for animal cells. They pretty much look like this. So let's start off in the middle. And in the middle of most cells, you have a nucleus. Now, the nucleus holds the DNA and the RNA. And we talked about this before, we talked about this last week. that the DNA is the blueprint of who you are. Deoxyribonucleic acid. And I believe next week we're going to talk way more about DNA. But just know that in this little thing here, the nucleolus, that has all the compacted DNA. And if you look at here, the chromatin, it's kind of less dense. You know, it's a little bit more spread out. But if you look at the nucleolus, it's really dense, like a rock in there. And remember we talked about hydrogen bonds? Well, DNA's got a ton of hydrogen bonds. And what did we notice about the hydrogen bonds? The hydrogen bonds were relatively weak. They had partial positive and partial negative attractions. And that helps the DNA to fold millions of times over and then coil themselves into this nucleolus. So what's a good, nice question? Where's the location of concentrated DNA? It's in the nucleolus. Where's the nucleolus? It's located in the nucleus of the cell, which is, it's like the computer. It's the brains of the cell. So the nucleolus here, right, that has a concentrated DNA. Now, when we need the DNA or the code within the DNA that tells the cell and tells the whole entire body what to do, and I'm not exaggerating when I say that, it tells the cell and your entire body what to do, what to produce. It has knowledge of when you're going to live, when you're going to die, and everything in between. That's why we were very, very interested in decoding DNA. And now we figured out the code, but we don't know what it means. And that was in the late 90s. And chromatin has DNA, but the chromatin is relaxed DNA, DNA that will encode and transfer onto RNA, which is like a messenger between the brains or the computer of this whole operation to the rest of the outside cell or the inside of the cell, and then of course to the outside world. And it's so important that it has its own skin. It has its own protective envelope, okay? So what could you do at home to learn how to identify this? Can you take this entire picture, put it on Microsoft Paint, right? And then erase all of these words and then A, B, C, D, E, F, G? Sure. Could you also look up, can you also look up an organelle quiz? That has one labeled. It's something like, you know, like this, and then with arrows. Sure, you could do that, right? And since you now know this is an anatomy class, you have to be able to identify things. You also know this is a physiology class. So you also have to know the function. So now you know the function of every one of these parts. The nucleus is to house concentrated and tightly packed DNA. The chromatin is loosely packed DNA. The nuclear envelope protects the nucleus, which is this entire purple pinkish thing. And what is the function of the nucleus? It is the brains, it is the memory, it is the boss of this entire operation. It dictates what is the function of the cell and how the cell is to operate, and how to live and how to die and everything in between. So do you think you can easily make that quiz? And therefore, right, come exam time, if you practice that, I can't guarantee that this picture is on there. Oh, who am I kidding? I can guarantee that this picture is on there because I'm talking about it in great detail. So it screams what? Hey, this is important. And I can make this, this photograph available for you guys on your, what is that thing called? On your announcements. Okay, let's move on to the next thing. Now, do you see these ribbons here? And they got little bumps. Well, that is your endoplasmic reticulum, specifically your rough endoplasmic reticulum. Your endoplasmic reticulum is a factory. It makes things. And the rough endoplasmic reticulum, also known as your RER, has these little bumps on it called ribosomes. And the ribosomes, they're kind of like factory workers that make protein. So the function of the rough endoplasmic reticulum is to make protein. Protein is made up of amino acids connected together by a peptide bond. Now, who told the rough endoplasmic reticulum exactly what to do? Well, the DNA here in the nucleus and chromatin has a code. The code gets transferred over through RNA, right? And then RNA then tells the rough endoplasmic reticulum exactly what order the proteins should be going, because that's the only difference between proteins is the order of the amino acids that make up a protein. So could I on an exam label this as like A, and then ask you what is the function of A, and then it's pointing at the rough ER, and you'll say protein factory. Or I could ask, what is this thing I'm pointing at? And that's the rough ER. Now, if you have a rough ER, right, the protein factory here, you know you have carbohydrates and lipids too, right? So there's got to be something that has to create carbohydrates and lipids. Well, guess what? That's this other set of like ribbons here, but they don't have any bumps on them. These have bumps, and the bumps are these little tiny ribosomes. This one has no bumps, therefore it's called the smooth endoplasmic reticulum. And this makes carbohydrates and lipids. And doesn't that make sense? Carbohydrates and lipids they're both made out of the same thing: carbon, hydrogen, oxygen. And what's proteins made out of? Carbon, hydrogen, oxygen, and nitrogen. Hey, doesn't that look like a good exam question as well? So you have smooth ER that has no bumps, rough ER that has bumps. Let's see, what else could we look at? And see, there's a little arrow here pointing at the little brown dots, and those are ribosomes, and they're protein factory workers within the factory that makes proteins. We also have ribosomes, well, here they are. We also have ribosomes that are also floating around, but we'll focus on the ones that are attached right here. Now, let's look at this thing. This green thing here that looks like it's like bubbling over, and then it has like little things in it, and it's bubbling over, and it's out pinching, you know, like these little things get sent out. The Golgi apparatus is the mailbox or the post office of the cell. It packages things, right? And you see it's creating these little bubbles. And these little bubbles, they could have anything like, for example, like this lysosome. Some means body, lyse means breakdown. They could be packaging these things to go digest something or to go eat something. This could also be packaging chemical messengers like hormones to be sent out into the cell, or neurotransmitters that can also be sent out into the world to communicate with other cells and other nerves. You could also create a peroxisome here. And just like the lysosome, we all know what peroxide does to a wound, right? It eats up all the dead tissue, and that's what peroxisomes do. They're kind of like garbage cleaners, lysosome as well. And also, this packaging is important because let's say, for example, you also have waste that has to be let go out of the cell. You don't want to be carrying garbage throughout your house. So you got to put it in where? Some sort of vehicle, some sort of bag. And this vehicle, the general term for this vehicle and this bag is a vesicle. And it can be secreted out, or it can, let's say there's glucose, it can be given to the other organs, because we're going to talk about this guy next. So the Golgi apparatus is the mailbox slash FedEx slash DHL packaging system that packages things not only for the protection of the cell, but also the communication of the cell to communicate things into the outside world, whether that be other hormones, neurotransmitters, or chemicals, or even waste, right? So that's the Golgi apparatus. Next, let's talk about this. This is the mitochondria. In the mitochondria, you have all these little channels, and within these channels, there's something called the electron transport chain that transports electrons from one molecule to another. Now, what does that do for us? The electron transport chain inside of this mitochondrion creates ADP. Let's write that down. If you guys can see it. So in the mitochondrion, which is the powerhouse of the cell, what does it do? It takes in, remember we talked about enzymes, and it takes in a substrate, right? It takes in like things and chemicals, and then it creates a product. Well, the mitochondrion takes in oxygen and glucose. And remember, the two really important fuel to be converted, to be metabolized in your body is oxygen. Therefore, we really, really need to breathe. And glucose, 6-carbon monosaccharide that we have to eat. And the mitochondrion will produce ADP. And ADP is, oops, not ADP, ATP. Adenosine triphosphate. Now, all it is an adenosine group with 3 phosphates attached to it. And we already know that bonds equal energy. Well, these are high energy bonds. And if you break ATP, it yields energy. And that's the metabolic fuel. That is the thing that keeps you alive. That's why the 2 things you need to require to stay alive and to have your cells reproduce and to thrive is both oxygen and glucose, or food, right? Because your body will break down food. So that makes total sense. And that's how your muscles move. That's how you're able to think. But the second you stop doing either one, what will happen? Things will start to fall apart, because who also powers all of this stuff? Remember we had bonds and things? There's energy that a computer requires, which is our nucleus. There's energy that has to keep these factories going. There's energy that's required to create these vesicles. And that energy comes from the mitochondria. Now, how do things move in and out of cells, and how do cells divide? We're going to talk about that a little bit in greater detail at another lecture. But you have these little scaffoldings within your cytoplasm. The cytoplasm is the jelly-like substance in between all of this. And you have these little scaffoldings, or these little highways, and they're microtubules. And at the ends of the microtubules, you have these things called centrioles. And the centrioles help separate the cell when it's time for the cell to grow and to divide. So if I ask you what these are centrioles. What is their function? Its function is for cell division. If I ask you what these things are, these little yellow lines, those are microtubules. Their function is for cellular communication / the structure and shape of the cell itself. And guess what the microtubules are made out of? Proteins. What do proteins require? Rough ER. Rough ER requires what? Energy from the mitochondrion. So when you're looking at this cell, this is like a little mini human being, right? It's self-enclosed, and it has a brain, and it's doing all of these functions. So if this cell has a problem at the DNA level, what's going to happen to all of these functions? It's all going to fall apart. That's why we're very, very interested in the DNA and the DNA code, and we're very interested in genetic diseases. Now, what's the other thing? We talked about lysosome, smooth ER, cytosol is the jelly-like, because we already know from looking at the microscope that all cells have different shapes and sizes, and the DNA predicts that and dictates that. But what maintains the shape are 2 things. The cytosol, which is the jelly-like substance in between all of this, right? That, I guess that's yellow brownish stuff. And of course, the microtubules that relay some sort of scaffolding / communication system within the cells. See, did I miss anything? Microtubules. We're going to talk about the cell wall in a minute. We're going to look at a better picture if I can find it. Nuclear envelope, nucleus. Okay, and this is mitochondria, and if you're wondering what this thing was. So let's now look at the plasma membrane. The plasma membrane is also known as the gatekeeper. It makes sure that everything stays in its place, and it selectively allows things in and selectively allows things out, hence the term gatekeeper. And I'm going to show you in greater detail what it is. Now, there's also protein extensions that could have microvilli and microfilaments, and this also helps the cell either in movement of the cell itself, or if it's lining maybe like something like the GI cavity, it also moves items up and down your cavity. For example, did you ever wonder how mucus gets coughed up? Well, on the lining of your cell wall, you have microvilli, and again, made up of proteins, and the proteins get, this microvilli acts like little fingers, and it moves the mucus up if there's excessive amounts of mucus. Then it causes the gag reflex and the cough reflex, and then you cough it up. That's why, a little side note, if you cough something up, do not swallow it. There's a reason why you have a gag reflex. There's a reason why you have a cough reflex. You need to get that thing out of the system, because what is the function of mucus? Mucus traps things. Mucus can even trap viruses. That's why coughing goes coughing, especially if it's a productive cough. Get rid of that stuff. And of course, don't spit into the street, because as we already know from COVID, I'm sure you heard and read about it, your mucus, when it dries up, it gets worse. So if you spit on the street, or you spit somewhere, or you cough somewhere without covering your mouth, those droplets, when they dry, they flake off and then they fly around. And then now whatever's in, because whatever got trapped in that mucus is now airborne. So we're always promoting to our patient, like, you cough into a tissue, you ball the tissue in another tissue, and then you throw it out and you wash your hands. You even wash your face if you could, if you can, right? And it's the same thing now. And you also have patients, some of your older patients, like my father-in-law, for example, they love their little handkerchiefs. Get rid of that. That's nasty. You know, you cough into that, and then you put it in your pocket, and then you bring it to other places. And remember, it goes, when the mucus dries up, whatever's left in there can survive 48, 72 hours. Tuberculosis can survive 72 hours. Most viruses, anywhere from 24 to 48. And then again, like I said, it becomes airborne. Okay? These little flakes become airborne. And again, yet another argument to wear the mask, right? Not necessarily for the virus, but for the other nasty things that are floating around. And the virus, when they're floating around on these little flakes, I mean, right? The flakes are very, very big. So, but the virus is very, very small. So if you wear your mask, right, it's a good way to have that extra filter. So this picture, right? I'm sure someone's going to ask me what's going to be on an exam. I can tell you right now. I'll be 110% honest. This picture in some form, this exact picture will be in the exam, and I'll just erase all of these and just write A, B, C, D, E, F, G, and then I'll ask one or two questions. I could ask, what is it? Or what does it do? Okay? So that is the cell itself. Let us look at the cell membrane. And there's a summary within your Carolina notes of the functions of each one of these organelles in your Carolina notes. And after I show you this next picture, I'm going to show you the location of those notes. So the next picture we're going to look at is the bilipid layer membrane. And let's look at that wonderful picture. And this is my favorite picture. I always use it. Copy image. And I'll make this these pictures available to you guys. God bless you, computer. Okay. Now the nuclear membrane and also the plasma membrane of the cell, we already talked about, they're both gatekeepers. The nuclear membrane protects the nucleus or the computer or the brains of the operation, but and also your plasma membrane, the outside wall, go back to this picture just to know what we're talking about. That's a cross section of this gray skin right here. And that is your plasma membrane, right? That is your cell, that's the cell wall. And in eukaryotic or human cell walls or animal cell walls, they're bilipid layers. And the bilipid layer has a hydrophilic head, sorry, right? So that likes water. So of course there's water on the outside and there's water on the inside. And you can see here, there's some microtubules going on here. And that's how we know this is the inside of the cell. So actually the cell membrane is a phospholipid bilayer. And we know that lipids or fats are not soluble with water. So it keeps the water on the outside, right? Because it has a hydrophobic middle. And it makes sense. If it's a hydrophobic fat layered middle, there's got to be cholesterol in there. And just know that cholesterol is just simply a transport protein for fats, so that fats can go in and out of the cell. And maybe you're familiar with some of these transport proteins. Maybe you go to the doctor and we talked about your VLDL and your LDL and your triglycerides, right? That's a measurement of fat. Well, your VLDL and your LDL, which stands for your very low density lipoprotein and your low density lipoprotein, those are just proteins that can carry, including cholesterol, that can carry fat going either into this, into the cell or out of the cell. That's why we like the high density lipoprotein. It's not really the bad cholesterol, but the high density lipoprotein is really just a vehicle to take fat out of your system. Now, the majority of the systems we have in our body likes to put fat into our system, right? That's why we harp on diet, and we also harp on exercise, because we want to always be in the middle and homeostasis. So cholesterol, you could see it's within the bilipid layer, and inside the bilipid layer is a layer of hydrophobic molecules, and on the outside is a layer of hydrophilic or water-loving. And that makes sense because there's water on the outside, water on the inside, but we have a nice lipid bilayer here to prevent things from going in and out. Now, you also have protein channels. Proteins are neat because they're made up of amino acids, and I want you to think of amino acids kind of like building blocks, like Lego. And depending on what Lego you choose, you can make certain things. So one of the things that proteins can make is a channel. Now this channel, they have doors and keys and locks. So this also shows how the phospholipid bilayer selectively picks things to go in and out of the cell. So let's say I have a whole bunch of waste here on the inside of my cell. Well, the protein channel knows that it's waste. It opens it up and then it throws it out. Another thing that these protein channels do is, do you guys remember the Matrix when Morpheus says all we human beings are is just batteries? Well, when you can think about it, we already talked about ions, plus and minus. And aren't batteries have a plus? Aren't batteries also have a minus side? And when you put the plus and minus together, you have what? Electricity. How do I know? I was one of those kids who took a 9-volt battery. If you put your tongue on the positive side of the 9-volt battery, you're fine, right? You put your tongue on the negative side of the 9-volt battery, you're fine. But what happens if I put my tongue on both the positive and negative side of the battery? Zap, I get electricity. Guess how your body generates electricity? These protein channels move ions like sodium and potassium and chloride, right? And you know that sodium is a plus, chloride is a minus. You put pluses and minuses together, right? You will have electricity, just like when you mess with a car battery, when you mess with the 9-volt battery. And you don't want to know my car battery story either. It is just as stupid, right? So these protein channels are very, very important. So you could see how protein synthesis, DNA, and all of it is connected, and they're selectable. Actually, in order to kill many antibiotics that kill bacteria, one of their functions is to kill this protein channel. If you kill that channel, can things go in and out? If things can't go in and out, the cell will die. But things must selectively go in and out. Another thing you also have in proteins are these integral membrane proteins. Now, this is a bilipid layer, and it's kind of like it's flimsy. But we know that skin or the outer layer of something has to be strong. So you have these integral membrane proteins to give the phospholipid bilayer some strength. Okay. Now, you'll also notice, remember I told you there's a lock and key mechanism here for the proteins, right? Well, the lock and key mechanism is based on chemical recognition of what the cell is and what the cell does. So you have these glycolipids made out of sugar and fat. Lipid is fat, right? Or glycoproteins, which is made out of sugars and proteins that do two things. One, it acts like a lock and key mechanism for these protein channels. Two, it also identifies the cell chemically. So based on the chemical structure of what this is how we can also identify what cells are. And if the cell is abnormal, right, or a pathogenic, don't you think it's going to have a glycoprotein coat or a glycolipid coat? How do you think we diagnose and hunt down HIV? We look for GP120 and GP95 and GP90. If we see that, we attach an antibody that hunts for it, and then we can find it. And that's called Western blot, and that's how you diagnose HIV. So when we're looking at this, I could ask you, what is this picture? And you'll tell me, oh, that's the gatekeeper of the cell. That's the phospholipid bilayer. Okay, these little round heads, are they hydrophilic or hydrophobic? They're hydrophilic because they love the water. How about the inside of this bilipid layer? Oh, that's hydrophobic. So it separates the inside water from the outside water. What is the function of glycoproteins and glycolipids? Does that look like a beautiful both A and B answer? It's for recognition chemically of what the cell is, or cell identification chemically, and it also acts like a door for your protein channels. What's the function of these integral membrane proteins? Oh, they're a structural protein to give the phospholipid bilayer rigidity. And what's the function of cholesterol? Well, cholesterol located inside the cell membrane, and it acts as a vehicle for lipids to go in and out of cell, because sometimes my cell needs lipid. What's inside lipids? Also known as fat, also known as adipose tissue. It's glucose. So if I invite lipids into my cell, that means I need the glucose, okay? Actually, that's also how cells get in. Insulin attaches onto a glycoprotein receptor. The receptor then opens up a whole bunch of doors or protein channels so that the big 6-carbon glucose molecule can go in. And that's what happens when you ate a heavy meal. Now, what happens when you haven't eaten for a while, you know, maybe around 6, 7, 8 hours? Well, glucagon comes in. We recognize glucagon, and then what's gonna be the signal for the cell? The cell then has to break down the fat that it stored earlier, to make glucose. And remember, why do we need glucose? The oxygen and the glucose go inside the mitochondrion, and the mitochondrion makes ATP, adenosine triphosphate, and that's the metabolic fuel that you need. So all of that is going on inside every nucleated cell in your body. Every nucleated cell has all the DNA information of who you are. So did we go over the cell membrane? Yes, we did. Cytoplasm and cellular organisms? Yes, they did. We're going to go over the types of cells in a moment. Now let's look. When you click on this, oops, let me be specific. It's in your task 3. Like, so if you go into task 3 and you click on cell structure and function, those are Carolina notes. Okay. And of course, everything that we went over, create a concept map to link cell structure, organelles, and the function of organelles. And that makes sense. And this set of notes can help you. And looky here. Let me go fast forward. Everything you want to know and everything that I just talked about. And looky here. Doesn't this look like a beautiful matching? Right? Don't you think you can go home, copy and paste this, and then, or take a piece, print this out, take a piece of paper, fold it in half, and then quiz yourself? Yeah, you could easily do that, right? What does the plasma membrane look like, right? Now, regarding the plasma membrane, I got to mention this. Osmosis. Now, what does osmosis mean? Now, here's an example on something called entropy, spelled E-N-T-R-O-P-Y. All the molecules in the universe want to spread out. That's why it requires energy in the form of bonds to keep things together. That's why you have to keep on eating and keep on breathing, or you will start to disintegrate. And that's exactly what happens when you die. When you die, the second your heart and your lungs stop and brain function and all that stops, you will start to deteriorate. Why? Because all your molecules want to spread out. And here's the analogy regarding osmosis. What happens when you go to Macy's or Nordstrom and you go into the perfume counter and the lovely lady or gentleman sprays you right there at the counter? At that moment, the concentration around you and that perfume bottle is high. But what does the molecules of that perfume bottle want to do? They want to spread out. So in a moment, what happens, right? The molecules then start spreading out from an area of high concentration to low concentration. That's why the further away you move away from that counter, you cannot smell or you cannot detect any more of those molecules from that perfume. And that's also what osmosis is. Osmosis is a flow of molecules from a high gradient to a low gradient, or a high concentration to a low concentration. This requires no energy. It's like rolling down a hill. You know, you bring your bicycle to the top of the hill. What happens? You don't have to pedal or anything. You just go downhill. And you're going from a high concentration to a low concentration. Another example is evaporation. You put water, a glass of water, in your living room. After time, you know, a couple weeks or whatever, the water is going to get less and less and less. Why? Those water molecules want to do what? Go from a high concentration, which is the water in the cup, to an area of low concentration, which is the rest of the air and the rest of the room. So that's what osmosis is. It's the movement or diffusion of water molecules from an area of a high concentration to low concentration. But the trick is, it's from an area of a high concentration to a low concentration through a selectively permeable membrane. Doesn't that look familiar? What is a selective permeable membrane that we already talked about? hey, this buddy right here. So that's how things move. So once these protein channels are open, if I have a high concentration of sodium out here and a low concentration of sodium inside, what's going to happen? Naturally, the sodium will do what? Go in, right? And when we detect that we have enough or we've reached homeostasis, there'll be a signal for the door to go to get shut. Or if you have equilibrium, which is the goal of osmosis, the concentration on the outside will equal the concentration on the inside. So now, when the concentration is the same, let's imagine that this is the semi-permeable membrane here, this little line. So you have room A, room B, room A, room B. Here is when we have homeostasis, when the right and left sides are in equilibrium, meaning to say is, let's say these little white circles is water, and these little dark circles are Kool-Aid, right? The amount of Kool-Aid is equal on both sides. The amount of water is equal on both sides. Therefore, both solutions that are in equilibrium are called isotonic. Another definition of isotonic is 0.9% sodium chloride solution, also called normal saline. That's why we put normal saline in our patients, because guess what the fluid of 66% of water that's in my body is made out of? It's made out of 0.9% sodium chloride solution. So NSS is isotonic to human beings, and it'll help my human being get into equilibrium. Now, what is hypertonic solution? Well, if you look at these two, room A, room B, separated by a semi-permeable membrane, do you notice that room A has way more Kool-Aid or solute, right? Or the powder molecules that get mixed up with the water to make your Kool-Aid. If you notice 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, there's an equal amount of water concentration on both sides, right? But this side has way more Kool-Aid or solute. If you have way more Kool-Aid powder and way more solute, that is called a hypertonic solution. And if you have way, way less, that's called a hypotonic solution. And of course, if you're in equilibrium, that is called an isotonic solution. And sometimes we play around with the chemistry. Hey, Belle, can you bring baby out of here? Sorry about that, right? If you have a hypertonic solution here and a hypotonic here, what's the body want to do with this semi-permeable membrane? Because of osmosis, it wants to get to equilibrium. Now, this is the reason why I say things like, wherever salt goes, water goes. So let's say this is a semi-permeable membrane. We already know that. And what happens? Through osmosis, right, you have a high concentration of powder, right, versus a low concentration. So where does the powder want to go? The powder wants to go this way, towards room B. And guess what? When it moves, it's going to take water with it. And that makes sense, because what do you think water pills are made out of? They're made out of salts. And then it tells your kidney to throw out salt. So if you throw out salt, what will follow? Water. And that's how water pills work. That's why hydrothiazides and diuretics, that's how they work. And if I remove water from your system, what's going to happen to your blood pressure? It's going to drop. And this is also why we don't like a high salt diet, because can you see here how the pressure here would be greater than the pressure here? Because this one has way too much powder. Now, if this was salt, it's going to draw water where? Into here. Remember, wherever salt goes, water goes, and this will have a higher pressure than this one. We want to be in equilibrium, and we want to be at the lowest pressure possible. And that pressure, which I just mentioned, the pressure of how much Kool-Aid is inside your little room or little jar there, that's called osmotic pressure. Okay? And we used to do this thing where we fill up these condoms with like sugar and we watch the sugar, but it never worked. And it was really, really messy, so we abandoned it. So what is the takeaway of those notes? Of course, not only the function, but also know what osmosis is, diffusion, and remember, osmosis is simply diffusion through a semi-permeable membrane. How does the diffusion go? It runs from an area of high concentration to low concentration. Wherever salts or solutes may go, what will eventually follow? Water. Okay, and it's the basis for diuretic therapies and blood pressure. And that's also the reason why, you should reduce your salt. Remember, food for fuel doesn't have to taste good, but it has to provide you oxygen and the glucose that is required for you to live. Now, let's do next week, cell structure, organelles, and function of organelles. Let us now go to discussion 3. What does discussion 3 look like? Now, you don't really have to watch the video, but it's nice. But you can just look up mitochondrial disease. Now, we already know what the mitochondria does, right? So relate that to what your knowledge of the mitochondria, right? Relate that to what goes, how it can create the disease and the pathology. And of course, how does the current management for mitochondrial disease, how does that relate to it, chemically, like the way we do it? Like, for example, the mitochondria creates ATP. So if my patient has no ATP, that means they'll have no energy. So how do we replace that energy? What can we do about it? So you're talking about two things. First, the definition and pathophysiology of the mitochondrial disease. That could be one paragraph. And then the second paragraph can be, okay, the current management for most mitochondrial disease is A, B, and C. And you can tell me the physiology of it. How does that work? And how does that help the mitochondrion create ATP? So that's due, of course, to at least 200 words. And make sure you got those citations, because all of you have really good rationales, and you're pretty good with writing, but I don't understand why you're afraid of citing things. It's going to be an issue in future classes if you don't get that squared away. Now, the bonding and properties of water, lab don't do. And you can look up scratch test, protein test, fat lab. We used to do that in the lab, but again, messy, didn't always work. So what is due next week? Case study. Click on the case study. Download the case. Wait, am I in the right? Download the case. Yeah, we already did this. Hold up. I think I'm in the wrong thing. Yep, I'm in the wrong thing. Lesson 3. My bad. And again, you have this nice video, and I think you could do quizzes on this. This is cute. And they're separate videos, and it's going through the same thing. Ooh, watch this. This is a nice little preview for next week's lecture. This is good, right? You could also look at cell membrane and more cell function, more notes on, don't have to complete the lab, but look at the notes. These are nice. So let's look at the application assignment. Download case. Now, of course, just like always, bring baby home. And it's going to look a little bit, answer, of course, this part 1, and the 5 questions. And look at this part here. We got a couple minutes, so we can talk a little bit about this. Okay. Now, the baby, of course, they already told you what the diagnosis is, and the baby has a genetic disease. And remember, genetics, your DNA is the blueprint of who you are. It says everything on what's going to happen and what disease you're going to have, what disease you won't. Now, answer these questions, but you're going to need to know in part two, you're going to need to know a little bit what this is. Okay? So, if you look at this, there's something called a genotype and phenotype. Genotype is this letter representation of what kind of genes you are. So you could be autosomal dominant, right? Which means that this big R, that means that's the thing that most likely will supersede the little r. So big R means that you're normal. The little red R means that you're going to have this disease, Leigh syndrome. Now, mommy and daddy here, they're both carriers. How do we know? Because phenotypically, their physical attribute is what? Phenotypically, they don't have the disease. Therefore, they're carriers. Now, they're carriers because genotypically, you have one big R, one little r. Now, this one little r is recessive. That means it's in hiding. Now, the only time little r comes out, if you have one little r from mommy, one little r from daddy, and then you can be affected. So this is a classic autosomal recessive inheritance, and this comes up on your NCLEX. So in the typical autosomal recessive scenario, which is the case of for this particular unfortunate patient, mommy and daddy were carriers. Because why? They had one recessive trait each. Now, what happens if they had kids? 1 in 4, or 25%, will totally be unaffected. 50%, or 2 out of 4, will be carriers. And 1 in 4 will have the full-blown disease. And that's called autosomal recessive inheritance. Okay? And I could take away all the people, right? And just have the R and little r. And then I could ask you, is this person, do they have the disease, or are they a carrier, or are they completely normal? And you will be able to tell me that all of these people are carriers. If I show you this, even without showing you the phenotype, you'll tell me what? Oh, this person has 2 autosomal dominant genes, a big R or capital R, capital R. This person is perfectly normal. This person with 2 autosomal recessive genes, this person has the disease. And in the case of this this little girl, it's Leigh syndrome. I could also ask you the other way. Let's not look at the letters. I could also ask you phenotypically. Phenotypically, this child is a carrier. What will their genes look like? Big R and little r. Phenotypically, what will because this person doesn't have the disease whatsoever. It'll be big R, big R. Phenotypically, this person has the full-blown disease, so it'll be little r, little r. And we have a question. Yes, sickle cell has runs by the same thing. You have carriers, but the thing about sickle cell is the carriers have sickle cell trait and may have some signs and symptoms, but the person who has full-blown sickle cell with crisis, that's the person's going to be affected, and that's the person who's going to be in significant amount of pain. And it makes sense because red blood cells, they carry oxygen and carbon dioxide. What happens if I don't get oxygen to where it needs to go? Then the mitochondrion can't make ATP, and that has the signs and symptoms of similar to mitochondrial disease. And the carriers have milder diseases, or they have milder forms. They may have just sickle cell trait. but full-blown sickle cell disease, it's debilitating. It's one of the few times that I'll give morphine to a teenager. I've had young men, 17, 16 years of age, doubled over in pain. And it makes sense also because remember, the red blood cell carries oxygen. What if I took away a third of your oxygen? You're going to start cramping up. Those of you who like maybe you're coming back from a workout in a long time and you overdid it, how do you feel? Everything hurts. Everything's crampy and you can't breathe. So do you have to memorize things to remember they are? No. And that's what these cases are for. So understand this. This could be a nice quick question on the exam. And know the difference between phenotypical, which is what you'll see physically, and genotypical, which what you'll see in the big R, little r. Oh, I'm sorry to hear about that, Miss Bass. And yeah, they do pass, and on the way, it's debilitating. Oh, I also missed another second part of the discussion page. Okay, let me go back to the discussion. But real quick, know this. This is good to know. And we'll be going over in different cases, different genetic stuff. So just do part 1 and part 2 because understanding the cell, oh, look at this lovely picture, right? Part 3, we already went into, oh, look at here. Do you think you could practice this as well? So just part 1 and part 2 for this case study. Okay, so part 1, bringing home baby, and part 2, the diagnosis. Leave part 3 and part 4 alone, okay? Because part 3, you know, it's already in your notes. It's more busy work than anything. Let us now, as per request, go back to the second part of the discussion. So I'm gonna go back to discussion 3. So what second part? Oh, the second part. So there's 2 parts. First, like, you know, in your introduction, you have to say, what is mitochondrial disease? What's its pathophysiology? But the second part is, what can be done about it? So you look up the management of what do we do for patients with mitochondrial disease, and you explain why does that make sense or why it doesn't make sense, right? But it should make sense. Because it should be geared towards replacing fuel, because we now know what the mitochondria does. It is the powerhouse of the cell. Okay, so first, of course, how mitochondria is affected, what's the pathophysiology, and what's the management? What can be done about the disease? And I hope that answers your question regarding the second part of the discussion. All right, with that being said, I think I covered everything. And if there's nothing else, you can add questions or you can unmute yourself. Is there anything else for this week? Other than midterms are in 2 weeks. This is week 3. Next week is week 4. I'll be putting out there a mock. Well, it's actually not a mock. It's a sample of an old exam that I had for this class. So it gives you an idea what kind of questions and how do I ask the multiple choice format for the exam. And again, you have the choice to take the exam online. It'll be done via Zoom and file transfer via Zoom of the exact exam. You'll have approximately 1 hour to complete the exam and to email it, email your answers to me. And of course, you must turn on your camera so I can see your face, so I know it's you taking the exam. And you know what? There's a million ways to cheat on this. But like I've always said, you cheat, the only person you're seriously gonna hurt is yourself. When you go out there and don't know what you're doing and kill people, they will prosecute you to the full extent of the law, and the hospital and the administration will enjoy every minute of it. I'm telling you, everyone I've ever known who cheated, and there's been a lot of people even in medical school who cheat. oh, what happened to them in their careers is just gosh awful. I don't wish that upon my worst enemy. So if we have nothing else to talk about, kindly put in your homework for week 2 by closing of business today, right? And I'll see you guys next week. Anything else? Anybody? Questions, comments, recipes?
Is there any strategy for, how can I say, like, it's a lot of information